Hello, this is Dr. Mamta. Let's study the types of hormones and their mechanism of action today. So, our objectives for today is to learn the classification of hormones, to learn the mechanism of action of hormones, and to learn the mechanism of action of second messengers. Okay, so when I say hormones, you will always be remembering the endocrinology, right? So, it's not only endocrine, there are many other things also which also release hormones. But for your information, from endocrine, okay? The endo is released from Greek words that is inside or within. The cranium, which means to separate or to distinguish. The term hormone, if we talk about, it was released from a Greek word that is hormaion, which means to set in motion, which describes the dynamic actions of hormones as they elicit the cellular responses and they regulate physiological processes through feedback mechanisms. Now, when you're asked to define a hormone, you can define it as a biochemical messenger released into the extracellular fluid by a cell or a group of cells which are recognized by specific high affinity receptors of the target tissues which regulate many functions of the body to maintain homeostasis. So, when I say neurocrine, it is neurocrine, which is a very easy thing. That is, the hormones are secreted or the chemicals that are secreted at the end of neuron, which is released into the bloodstream. That is neurocrine. When the hormones are secreted by an endocrinal gland into the bloodstream, that is endocrine. When the hormones are secreted by a cell and that acts on its adjacent cell itself, then that is called paracrine. When a hormone is secreted by a cell and that affects that own secreting cell, it's called autocrine. Now let's study the classification of hormones. We can classify them based on their chemical nature or based on their mechanism of action. Firstly, to classify them based on their chemical nature, they can be either amino acid derivatives or a small neuropeptide or a large protein or a steroid hormone or a vitamin derivative. Examples for amino acid derivatives are dopamine, catecholamine, thyroid hormone. There are many other, I'm just giving you a few examples for each. Small neuropeptides can be gonadotropin releasing hormone, thyrotropin releasing hormone, somatostatin, vasopressin. Large proteins example would be insulin, luteinizing hormone, parathyroid hormone. Steroid hormones examples are cortisol, estrogen. Vitamin derivatives are vitamin A and vitamin D. So we can also classify hormones based on their mechanism of action. So when I say mechanism of action, it is based on how are they going to act, right? Now, how can you imagine how they are going to act? You can either imagine a cell on which it is going to act, for which it has to bind to something, and that can be a receptor. And that receptor can be on the cell membrane, or it can be in the cytoplasm, or on the nucleus, or in the nucleus on the DNA, right? So, based on that, we divide them. Firstly, the hormones that bind to the intracellular receptors, example, androgens, calcitriol, estrogen, glucocorticoid, mineralocorticoid, progestin, retinoic acid, thyroid hormones. When I say intracellular, it can be either cytoplasmic or it can be nuclear receptor that it binds to. When I say cell surface receptors, so hormones are going to bind to the cell surface receptor, but it is going to act. So, if it has to act in the cell, then that means there should be some other chemical which is coming and acting for it, right? So, that is the second messengers. There are multiple second messengers like cyclic AMP, cyclic GMP, multiple, okay, calcium, calcium calmodulin, all that. Okay, so firstly, that which binds to the cell surface receptor and acts through cyclic AMP. Examples can be catecholamines, HCTH, ADH, calcitonin, human HCG, that is human chorionic gonadotropin, corticotropin releasing hormone, FSH, LPH, that is lipotropin, the glucagon, luteinizing hormone, melanocyte stimulating hormone, PTH, somatostatin, and TSH. Next, hormones which bind to the cell membrane receptor and act through cyclic GMP are atrial natriuretic peptide or factor and nitric oxide. Then another hormones which bind to the cell membrane and act through calcium or phosphatidylinositols or both could be acetylcholine, alpha 1 catecholamines, angiotensin 2, vasopressin, cholecystokinin, gastrin, gonadotropin releasing hormone, oxytocin, platelet derived growth factor, substance P, thyrotropin releasing hormone. There can be hormones which act through kinases or phosphatase cascade. Examples can be adiponectin, erythropoietin, fibroblast growth factor, growth hormone, insulin, leptin, prolactin. Okay, so let us study the actual mechanism of action. So to study the actual mechanism of action, as I said earlier, they need to reach the target tissue, right? See, they are either secreted by an endocrine gland or a neuron or a paracrine cell. But for it to act, it has to reach its target cell first. Now, after reaching its target cell, it has to either bind to its cell membrane receptor or the cytoplasmic receptor or the nuclear receptor, right? So then they're going to form the complexes with the receptor, right? So what is that secondary response which is going to be obtained following the receptor hormone interaction? It can be through G protein coupling or through activation of enzyme activity within the cell or through direct activation of transcription. So firstly, let us study the G protein coupling. So till now, where are we at? We studied what is a hormone, we studied the classification of hormones. Now we are studying the mechanism of action of hormones, wherein the hormones are released by a gland or a neuron and are released into the bloodstream. Now they have reached a target cell. They are now bound to a receptor. Now, after binding to the receptor, how are we going to get a response? How? Firstly, it can be through G protein coupling. So, the coupling of receptors with groups of cell membrane proteins, which are called, GMP or G proteins, that is guanosine triphosphate binding proteins. So, what are these like? They are G proteins are the 7 transmembrane segments that loop in and out of the cell membrane. This part of the receptor extends into the cell cytoplasm and that is coupled to three parts, that is alpha, beta and gamma subunits. I think in this diagram it becomes more clear to you all. So this is the cell membrane. You know that it is fluid mosaic model, which is more followed for the cell membrane, right? So here this is the G protein. Okay, G protein coupled receptor. So this is the G protein and the G protein coupled receptor. As you can see, there are seven like that is the the receptor loops in 7 times through the cell membrane, right? And that is attached to the G protein, which has got alpha, beta, and gamma subunits. Now, the hormone was released, let us say, for example, it was released by some endocrine gland. It has reached its target cell. Okay, so it's binding to its receptor, that is a G protein coupled receptor. now, now, once the hormone binds to the G protein coupled receptor, can you see in this when the hormone is not bound, the GDP is away from this cytoplasmic part of the receptor, right? Now, once the hormone binds to the receptor, you can see that the G protein gets activated, okay, and the alpha subunit binds to the heteromeric loop of the G protein, right? So once that gets, when that is nothing but the activation of G protein. So initially it was GDP and then it becomes activated, wherein the GTP activated alpha subunit. Hence, gamma and beta subunits get separated, and now this activated alpha, that is alpha activated G protein, will now bind to the target enzyme which will bring about the actions of the particular hormone which is bound, right? So here you can see, this is again an example, again the same thing, but you will see what is going to happen next. Okay, so hormone bound to it, then the inactive G protein binds to the transmembranous heteromeric receptor, then that is activated, the G protein is activated and alpha subunit gets separated. This binds to multiple things which will bring about the actions of a particular hormone. Like for example, let us say um, any hormone which is bound to intracellular receptor. Okay, its actions, like for example cortisol, the functions of cortisol are about lipolysis, the proteolysis, and then increasing the glucose levels in blood. How is all this being brought about? By by through different actions within the cell, right? So that was an example, but G protein coupled hormones are different. Okay, so when that when that particular hormone is binding to the cyclic GMP or the GMP, okay, so activated GTP is going to bind to the potassium channel, open the potassium channel, and hence potassium goes out. So whenever the potassium goes out of the cell, it is going to, I mean, that particular action which is related to that cell is going to be brought about. It can also activate other enzymes like formation of cyclic AMP or formation of cyclic GMP, which are the second messengers through which the actions are brought about. Or it can bind to the nucleus, bringing about gene transcription and releasing proteins which bring about required actions of a hormone. Or they can activate enzymes within the intracellular area which will bring about the particular activity of that hormone that which is required, right? So best example that which we can take are histamine. Okay, so the G protein coupled receptors represent approximately 40% of all the drug targets in the marketplace. For example, H1 receptor antagonists are used in treating allergy, which for example is cetirizine. Ranitidine is a H2 receptor antagonist, right? So we use it to treat gastritis. So that was about the G protein coupling. So we studied what is or how a G protein looks like, and then we studied how a hormone binds to the G protein coupled receptor, and then how is the G protein being activated, and then how are the different actions of hormones being brought about. Now, another mechanism of action was to activate or activation of enzyme activity within the cell. How is that going to happen? Now, an enzyme linked receptor, which is also known as a catalytic receptor, can be a transmembranal receptor, right? So where the binding of the extracellular ligand, which is nothing but a hormone, will cause enzymatic activity. That is, once the hormone binds to the receptor, there will be direct activation of few enzymes which will bring about the necessary actions of that particular hormone. For example, is we call it Janus kinase and stat Janus Jack stat complex. Okay, so the best example here could be prolactin, right? So the Janus kinase, Jack is Janus kinase. It is bound to the signal transducer and activator of transcription complex, that is called JAK-STAT complex. Okay, now you can see that Janus kinase is a part of the receptor which is bound to the cell membrane. Okay, you can see that. But stat is present in the cytoplasm. So before the hormone binds to it, they are all inactive, right? They are not bound to each other. Now here you can see that once the ligand here is nothing but the hormone, binds to its receptor, you can see that the Janus kinase gets phosphorylated, right? And then that which will attract the stat, that is signal transducer and activator of transcription, Now, all these, the receptor, the genus kinase phosphorylated part, and the phosphorylated stat is nothing but genus kinase or JAK stat complex. Now, this is activated. Okay, so when the stat complex is phosphorylated or is activated, it can now enter the nucleus where it binds to the DNA and brings about transcription, and hence the required proteins are formed out of translation, and then those proteins will bring about the required actions of that particular hormone which is bound. Okay, so the best example here can be prolactin and growth hormone. Okay, so I mean, not only, see, here I said that the hormones are going to bind to the cell membrane and cause the activation of enzyme inside, right? So one example that I gave you now was Janus kinase mechanism. Like that hormone binding to the membrane can activate other enzymes also, like adenylyl cyclase and guanylyl cyclase, right? So they form cyclic AMP and cyclic GMP. These two together will now help in what is called as the activities or form the actions of that particular hormone. Okay, so let us study the mechanism of action of hormones, the third part, that is through the direct activation of transcription. Okay,